Hey everyone, this is Dr. Arun Kumar K. Today I will discuss about structure and functions of the insect integument. So you can see in the picture. So in one part of the picture you can see the suit of armor, and another side you can see the exoskeleton of an insect. In inside, so you can see the exoskeleton. So usually in any organism, it may be human beings or it may be any other mammals, you can see the endoskeleton, because so we are having a skeleton inside our body which is giving a proper structure and strength to our body. In the same way, in case of inside, instead of endoskeleton there will be exoskeleton. So you can compare with the suit of armor. You know that where suit of armor used normally to in the war field to prevent the injury from any other people. Is it not any other warrior? In the same way, it is going to give the protection from the any mechanical injuries, or maybe even it is giving some proper structure. Now in detail, let us discuss about the integument or maybe body wall of an insect. The body wall or integument of an insect forms an exoskeleton covering of the insect body. So and it forms a composite structure which forms the skeleton of the insect body. It provides area for muscle attachment and protects from desiccation. Means so loss of moisture is going to be avoided or prevented from because of exoskeleton, and physical or mechanical injuries also going to be prevented, and gives the perfect shape and strength to the body and its appendages. See, insect integument consists of totally three layers. The lowermost layer is inner basement membrane, and second is the middle epidermis or hypodermis, and the topmost layer is cuticle. So you can see the structure of an insect over here. At the lowermost you can see the basement membrane. This is the basement membrane. So and above, just above the basement membrane, you can see the epidermis, or it is also called as hypodermis, where you can see the dermal glands, so which is going to produce the dermal is what you call secretions over here. Next, trichogen cell, which is going to produce the trichos or air, which we can see outer surface of the insects. So next to turbogen cell, it is going to form the cetal socket and gives the nourishment to the trichogen cell actually. Next, if you see the cuticle, again you can see the different parts are there here: exocuticle and then endocuticle. Exocuticle, you can see it is highly pigmented layer, is somewhat darker as compared to the endocuticle. At the topmost layer, you can see the epicuticle. Okay, so in detail we are going to discuss later. Okay. So first one, basement membrane. It is the basal part of the body wall formed from degenerated epidermal cells and appears as a non-living amorphous granular layer of the integument, because there will be no cells or there will be no living cells present in the basement membrane. That is why it is called as non-living cells or non-living layer of the integument. So if you see the measurement of this basement membrane, hardly 0.5 microns in thickness and consists of fibrous proteins, glycose, aminoglycans, so okay, and which are polymers of disaccharides. And the basement membrane forms a continuous sheet beneath the epidermis, where muscles get attached and become continuous with the sarcolemma of these muscles. And second layer is, or middle layer is epidermis. It is a unicellular, you can see only single cell be present, unicellular layer formed from the polygonal cells, which modifies into cuboidal or columnar during the process of molting. So these cells consist of a well-developed nucleus and other cytoplasmic contents. So adjacent epidermal cells are also held together by means of certain cytoplasmic process. It is called as desmosomes. So here all the epidermal cells are usually granular, and they are going to secrete cuticle and then enzymes involved in production and digestion of the old cuticle during the molting. And what do you mean molting, we are going to discuss later. The epidermal cells get differentiated into the following types based on the function they perform and they modify into. So first one is dermal glands. They are going to produce the cement layer. So cement layer is going to give the protection from any desiccation or any physical injury or anything. And trichogen cell, that is going to produce the hair-like structure or seta, which we can see outside of the integument or the surface of the insects. And molting glands secreting molting fluid which digests the old cuticle. Actually molting means it is nothing but the shedding of old cuticle followed by the formation of the new cuticle. Okay, that is going to be digested by this molting gland. And peristigmatic glands, which are present around the spiracles in case of diptera larvae. Okay. So next, cuticle. So it is the outermost thick layer of the insect integument secreted by epidermis, and it is divided into again two regions, that is upper epicuticle and inner will be procuticle. Okay. So epicuticle, so it is a thin and outermost layer varying thickness from just 1 to 1 to 4 micron. Chitin is absent here in the epicuticle, and it consists of the following layers. So usually in JRF examination or anything they are going to ask, chitin is absent in which of the following layers. Okay, so in epicuticle chitin is absent. So again you can see the 4 layers in case of epicuticle. There is a topmost one is cement layer, next wax layer, next polyphenol layer, and fourth one is cuticle layer. So cement layer it is secreted by dermal glands, and it is composed of lipoprotein. It composed of lipoproteins, and it protects the body from external damage. So just like a cement layer on our walls in the home and all. Okay, wax layer it is a prominent layer and 0.25 micron in thickness and consists of long chain hydrocarbons and esters of fatty acids and alcohols. It serves as a waterproof layer preventing water loss or maybe desiccation from the body. Next, polyphenol layer. It is a non-static layer containing various type of phenols, and that's why it is called as polyphenol layer, which are mainly used in the formation of the proteins, and it is resistant to acids and then organic solvents. That is the reason even if you are going to dip the insects into the any acids or any organic solvents, they are not going to be get dissolved. So cuticulin layer, fourth one. So it is a amber color thin layer over the surface of the epidermis, which is strengthened by outer polyphenol layer, and it serves the purpose of permeability and also act as a growth barrier. Okay. Next, procuticle. So again this procuticle is different into exocuticle and then endocuticle. So after let's discuss one by one. You can see in the picture over here. So this is exocuticle and this is endocuticle. Okay. Okay. So exocuticle, it is darkly pigmented, hard, sclerotized, means very hard one it is. It offers rigidity to the cuticle and consists of mainly chitin and a hard protein which is called as sclerotin. So because of sclerotin only, they are very hard. And endocuticle, it is very soft, light in color and unsclerotized because there will be no protein called as sclerotin here. It contains more chitin but lacks hard protein, that is hard protein, that is sclerotin. Okay. Next, pore canals. There are numerous fine vertical channels, transverse both exo and then endocuticles, measuring about less than 1 micron, that is 0.1 to 0.15 micron in diameter. You can see in this picture here, the linings which we are seeing here, they are nothing but the pore canals. Okay, they run perpendicularly from epicuticle throughout the length of the cuticle. They are useful in transportation of the cuticle material and then enzymes to the outer pore and then epicutical parts. Okay, so totally there are two major components of the insect cuticle. There is chitin and then proteins. So first one is chitin, which is very important in case of insect integument. It is a nitrogenous polysaccharide. It accounts for 25 to 60% of the dry weight of the cuticle. So the chitin was named by Audier in the year 1834. It consists of high molecular weight of a polymer of anhydro N-acetyl glucosamine. You can see shortly it is called as NAGA, N-acetyl glucosamine. So which is joined by beta glucosylate linkages. So it is embedded with the proteins in the procuticle and forms glycoproteins, and it is insoluble in water. That's why even if you dip into the water also, it is not going to be dissolved. Alcohol, organic solvents, dilute acids, and concentrated alkalis, but rarely. It is soluble only in concentrated mineral acid and then sodium hypochlorite. And the specific gravity of this chitin is 1.4 weight by volume. Okay, and other proteins also present along with the chitins in case of insect integument. They are arthropodin, sclerotin, and then resilin. So first one is arthropodin. It is a soft water soluble protein present in endocuticle, and the conversion of arthropodin into sclerotin is known as sclerotization or maybe tanning. And sclerotin, it is also called as tanned protein because the because the light color convert into hard color and maybe dark color here. So which is amber color and present only in exocuticle, not present in the endocuticle. And resilient, very important, many times they ask in the examination. It is a rubber like elastic protein which is colorless and present in the joints such as wing hind ligaments, leg joints, or clypeolabral joints, or sutures, and even tergosternal joints. So if you see the cuticular or maybe integumental modification in case of insect integument, so it is modified into external outgrowths or maybe internal invaginations. Okay, so outgrowths, if you see, there are two parts again, two divisions or two kinds are there: cuticular appendages and then cuticular process. Cuticular appendages means there you can see the presence of the membranous articulations, and in case of cuticle process, there will be no presence of any membranous articulations. And again, cuticular appendages can be classified again into setae and then spurs. In case of setae, you can see only one cell, that is why it is called as unicellular in origin. But in spurs, you can see many number of cells, that's why it is called as multicellular in origin. And invaginations, inside growth of the insect integument, there is apodems and then apophyse. Apodems usually they are hollow invaginations, and apophyses are solid invaginations. Let us discuss one by one. First one, cuticular outgrowths. They are divided into cuticular appendages and cuticular process depending on the presence or absence of the membranous articulations. So first one is cuticular appendages, where you can see presence of membranous articulations. They are the outgrowths of the cuticle are integument connected with it by means of membranous joint. Okay, so they arise from modified epidermal cells, and again you can see different classification, there is seta and then spurs. So first one is seta. It is also called as macrotrachea because macro means bigger one, mega. Trachea means hairs. They are usually commonly known as the hairs arise from the cup leg as well as our feet. You can see in the picture. So they are usually bigger in size. Trachea means they are hair-like structure. So setae are hollow structures developed as the extension of the exocuticle and are produced by a single enlarged epidermal cells which is called as trachogen. And articular membrane is usually produced by a second epidermal cell which is called as tarmogen cell. You can see in the picture over here. So setae have a role of axonic importance and vary with the species to species. That is the reason the arrangement of these setae or hairs in the insect can be used in the taxonomy. The study of arrangement of setae is known as ketotoxy. Many times they ask in the examination. Ketotoxy is nothing but study of arrangement of setae in the insects. Okay, so it is going to vary from one species to another species. There are different kinds of setae. So first one is clothing hairs, next scales, third one is glandular setae, and then fourth one is sensory setae. So first one is clothing hairs. Clothing hairs means just like a cloth covered throughout the body. These are hair-like structures that cover the entire body, legs and then wings. You can see examples of any pollinators because they are going to collect the nectar. At the time of collecting the nectar, you can see their pollen is going to get attached to this one. Okay, so honeybees is the example, or any other pollinator. Next is scales. They are these are varyingly pigmented plate-like structures covering the body as well as the wings of adult moths and butterflies. Lepidoptera. Lepidoptera means usually scales. Okay, so it is nothing but outer growth of an insect integument. Okay, next to glandular setae. So I think in from childhood and all you are familiar with this one. So in most of the caterpillars, the Lepidoptera it is present. So setae that functions as an outlet for the secretion of epidermal glands. If they are stout, they are called as glandular bristles. In case of certain larvae of Lepidoptera, that is going to produce the poisonous gland. Next, sensory setae. In the name itself, these setae are the hairs are sensory function. So these are the setae or trichomes provide with the sensory receptors or sensory nerve cells and respond to the external stimuli. Okay, these are present on the antenna or leg and then mouth parts. Next, spurs, second one. So occurs on the legs of many insects and differ from the setae in being multicellular in origin. In case of setae, only one single cell will be present, but it is not in case of spurs because you can see many number of cells. Okay, and cuticular process is the second classification. They have no membranous articulations. They are fixed to any particular part of an insect. Again, there are two types. One is microtrachea or fixed trachea or oculi. Second one is spines. So microtrachea, in the name itself, that they are very small in size. These are minute hair-like structures found on the wings of Mecoptera and then certain Diptera, but not present in all the insects. And spines, so they are the outgrowth of the cuticle, which are more or less thorn-like in structure and they are immobile. So cuticular invaginetions, if you see, there are two, apodems and then apophyses. So apodems, they are hollow invaginetions which provide area for muscle attachment, and apophysis is nothing but it is a solid invaginations of the cuticle which gives a mechanical support to the various organs with forming a distinct skeletal structures. Okay, so you can see the picture over here, apodems and then apophysis. Apophysis is also called as fragmenta in case of apodems. Okay, so you can see the gap over here, it is a halo invaginations. Okay, just like other integumental part, you can see the cuticle vertical basement membrane can be seen here, and epidermis, cuticle, again you can see epicuticle and okay, and fragmenta or apophysis you can see completely filled. Okay, solid invaginations. Okay, so this is about the insect integument what I wanted to discuss. Thank you.